So this is a typical picture of the muscular artery. The size is way smaller than the elastic artery, in fact. And again, we can simply see this nice simple squamous epithelium, the endothelial cells, and this is the internal elastic lamella. This is the border between the endothelial layer and the tunica intima and the tunica media. And this is the tunica adventitia, where we can find the loose connective tissue, and the vasa vasorum right here, the small vessel. The question is, how do we distinguish histologically in the macroscope between the artery and the vein? Well, this has to do with at least two different components. One, of course, is the histological description that was discussed before, the difference between the tunica intima, the tunica media, and the tunica adventitia. This is the easiest thing to see in the thickness ranging from the two different vessels. Another one is that typically we'll see the veins that have a more irregular shape, not the typical circular, nice, round, spherical shape. In the case, for example, this is the ideal example. This is the muscular artery and this is the muscular vein. Of course, they're both in the same range in the thickness, but nonetheless, we can see that the shape of the vein is way more irregular than the more regular shape of the artery. And of course, if we zoomed in, we'll be able to see that the tunica media is almost non-existent, very, very small, very, very thin in the vein, and very, very thick in the artery, such as in this case. So this is another special stain where we can actually see the elastic fibers. And this stain, actually the Verhoeven stain, serves the purpose of visualizing the difference in the thickness of the tunica media and the contents of the tunica media. The tunica media in the aorta, for example, is filled. It's purely black and brown pigments, and this is because we can actually visualize all the elastic fibers inside the tunica media of the aorta. In the vena cava, we see again the colorimetric differences are obvious, and they're way less prominent. So this is again the classification. We're going to find this classification between the elastic arteries, muscular arteries, small arteries, arterioles, capillaries, venules, small veins, medium veins, and large veins. This is a size-based classification. Typically more than 10 millimeters is the elastic, 10 to 1 millimeters is the muscular, 0.1 millimeters to 1 millimeter is the small, and so on and so on and so on. Now, we should discuss in this case the differences between the capillaries and the rest of the vessels. Now, we already discussed about the tunica intima, tunica media, and tunica adventitia. In this case, we should focus just a bit more about the function and the structure of the capillaries, because the capillaries are actually the most active and the most important structures for diffusion and for exchange of gases, exchange of nutrients, exchange of molecules, exchange of hormones. Generally, this is one of the most important part of the system. In fact, this is why we have high blood pressure in the arteries and low blood pressure in the veins, exactly for this, to have the balance between hydrostatic pressure and osmotic pressure, which of course facilitates the proper exchange of all the molecules from the blood to the interstitial tissue. So let's zoom in the structure and see what it is. We're going to find again the typical endothelial structure with the typical endothelial functions, and everything is going to be exactly the same as in all of the other vessels in the human body. The major difference is that we find here a unique cell. This is called a pericyte. Now the pericyte is a sort of stem cell, an undifferentiated cell with specific contractile functions. Now the function resembles that of the smooth muscle, and in fact, whenever we have the angiogenesis or vasculogenesis, the pericyte will differentiate and mitotically divide to produce the smooth muscles. So the pericyte should be considered both as a functional contractile cell, a precursor cell of the smooth muscle cell, and also of course as the stem cell that actually will give rise to the tunica media in case of the proper function, the proper growth factors being exerted and expressed in these areas. Again, this is the system that I was talking to you before, and the whole point of the maintenance of the high blood pressure is to have a high hydrostatic pressure from the arterioles, and of course the osmotic pressure, which is mostly controlled by the albumins and other components inside the blood. So again, this is why we need to have a higher blood pressure in comparison to the vein, not generally high, just comparing between the arteries and the veins. There are many different conformations and confrontations of the macrovasculature. This is how the systems of the capillaries are actually structured and control specific functions. We have, for example, in cases of whenever we're in cold or exposure to cold or exposure to blockades and so on and so on, there are many systems that we can actually escape to and actually save heat and save in different procedures. For example, let's take the example of the cold. In the case of the exposure to cold, we're going to have the activation and the contraction of the pericytes. So of course there's going to be less blood going to this area, and because the blood is the primary form of heat in the human body, that it actually moves and let's say transfers heat from one place to the other, is of course the blood. In this case we save loss of heat, and this is mediated by the constriction and the very aggressive, let's say, constriction of the pericytes. And this is exactly why we have cold fingers when we're exposed to cold, because the capillaries of our fingers, of our fingertips, are completely shut, vasoconstricted, in order to prevent heat loss and to maintain as much heat as possible. Now this is one of the examples. Another example is the portal system. What is the portal system? I'm sure you've seen this system, the main portal system in the liver and many other structures, also in the hypothalamus and so on and so on. Well, what is the purpose of the portal system and what is it? Well, this is the typical simple pathway that we have in this, let's say, in a specific organ or a specific location. The arteriole bringing the blood, the capillary exchanging and diffusing all the proper nutrients and so on, and of course the retrieval of the toxic byproducts and the deoxygenated contents and blood from the vein. Well, again, the most important thing to remember is that the surface area of the capillaries is directly proportional to the amount of molecules that come and go from the blood into the interstitial tissues. What does it mean? It simply means that the more surface you have, the more active surface you have for the capillaries, the more molecules you can exchange from the tissue and the organ to the blood and vice versa. So, for example, when we take in the system of the liver, okay, one of the primary functions is the detoxification. And this is, for example, in this portal system right here, practically doubles the surface area, and increases the surface area. So there is way more exchange of gases, the exchange of molecules, way more exchange of active and products and that are nutrients and so on and so on. Well, the point again of the portal system is to increase the surface area and as a consequence serve the function of the organ or tissue way better than if just to have the simple pathway. So let's move on. There are different capillary types, and this is why. Well, because different organs have different needs. For example, we already saw this example right now with the liver, for example. Now, the most common form of capillary is the continuous capillary. And there are these three two different types of capillaries are based on the differences in the architectural differences between the basal membrane and the endothelium. Well, the continuous capillary is nothing more than just a simple endothelium extending throughout the whole capillary and a continuous basal membrane that fully covers the whole diameter and the whole external area of the system of the endothelium. And this is for a normal, most of the human body actually contains continuous capillaries. Now, there are two different ones. There is the fenestrated capillary and the sinusoid. For example, the fenestrated capillary means what? That the endothelium actually contains fenestrations, small little holes that allow for the increased exchange of nutrients and molecules from the area of the lumen to the interstitial tissue. In other structures, in other organs where we need to actually exchange way more, even cells, even way bigger molecules, proteins, and structures that will not normally pass through the fenestrations of the fenestrated or through the continuous capillary, in cases where we need to have the maximal exchange of sized molecules, of big and proper molecules, this actually is being served, through the sinusoid or discontinuous capillaries. A typical example is that in the spleen. The spleen contains high amount of discontinuous capillaries, and this is because this is the location where we have the filtering of the blood, and we actually clear out the old erythrocytes. So erythrocytes, because they're big in size, they're not able to pass through the fenestrations or continuous basement membrane. So in this case, for example, in the spleen, we have the sinusoids, which allow for the full movement of big molecules and big even cells in this case. And we can see the discontinuous and big pokes and big holes from the fenestrations, from the basement membrane and the endothelium to the tissue that actually is necessary to receive or provide such cells or such large molecule. Let's talk a bit about the lymphatic vessels, and then we're going to see the structures in the microscope. So the lymphatic vessel, again, the definition of lymph again is the excessive amount of interstitial fluid coming to the specific area of the tissue. As we saw before in this picture, we have the hydrostatic pressure and the osmotic pressure. Because there is a difference in these two pressures, hydrostatic is always a bit more and a bit bigger than the osmotic pressure. There tends to be a higher amount of deposition of fluids and contents towards this tissue, and of course all this excess tissue, all this excess fluid is called lymph. So it's the role of the lymphatic vessels to reabsorb all this extra interstitial fluid and of course empty them in the end in the vein system, the venous system. Let's see the tunica intimas, medias, and adventitia inside the vessels. So here we have the elastic artery and large vein. This is the part of the tissue that actually we can see the large artery. In comparison, we should see the thickness of the large vein. Now, the diameter, of course, in both cases is typically the same. And we can see the difference in the thickness, and we can see the difference in the colors and the difference in the structures. So, let's actually visualize the whole structure. First, we have the tunica intima, exactly, which is this small layer, this very small layer, which actually is covered by the tunica, the endothelium. And this is, again, we discussed the function of endothelium, and this is the simple squamous epithelium. This is the endothelium. This tiny layer right here is the one that actually is the subendothelial layer. This is just a small layer that contains collagen fibers, and in rare cases we can actually see one or two free connective tissue smooth muscle cells. Now, exactly beneath we can find this, let's say, components, these structures are the elastic lamella. These are the elastic fibers that are produced from the smooth muscle cells that cover up throughout the whole layer of the tunica media. Again, we can see layer of elastic lamella, elastic fiber, smooth muscle right here, and again and again and again. And this different interchange between the smooth muscles and the elastic layers, elastic lamella, allows for the two functions to occur, both for the vasodilation to actually occur, because we have the elastic fibers that allow for this the outward pushing and the outward opening of the dilation of the vessel, and we have the vasoconstriction, which is mediated by, again, the smooth muscles right here, which constrict all together as a unit. Now, if we move on further on, we can actually see this is the border between the elastic, the tunica media, and the tunica adventitia. The tunica adventitia, as you can see, is filled with connective tissue, many fibroblasts interspersed, and we can also see the nice and clear adipocytes right here. Now let's go to the vein. So this is the vein. This is specifically this is the brachiocephalic vein. Now, if we zoom in, this is the lumen again. We can zoom in the tunica intima, and we can see the same exact structures. We can see very again the simple squamous epithelium and the very short and small subendothelial layer. Now, all of this, just this part, sorry, only this part, this is the tunica media. This is the very undeveloped and underdeveloped tunica media with very few smooth muscle cells, way low amounts of elastic fibers, and of course this is not the point. This is not the point of the vein to compress blood or to constrict the blood, just simply to carry it back to the lung and the heart. So of course the need for the tunica, for a large and thick and prominent tunica media, is not evident and is not actually present. But we do see a high amount of a very large and very thick tunica adventitia again. And we can also find the vasa vasorum. Let's actually zoom in and find one. There you go. This is the vasa vasorum. This is the vessel that allows specifically that actually provides nutrients and blood to the rest of the tissues, the fibroblasts and so on.